this is module number 4, which is related to non-clinical or preclinical writing. So first, we'll discuss about clinical and non-clinical writing. Okay. So preclinical studies. So we all know what are preclinical studies. Preclinical studies are the studies which are conducted on animals before the commencement of clinical trial process on humans. So preclinical development, also known as preclinical studies or non-clinical studies, is a phase of drug development that starts before clinical trial testing on human. Okay, so preclinical development, it is also known as preclinical studies or non-clinical studies, and it is a phase of drug development that starts before the clinical trials, that is testing on humans, and is characterized by the collection of crucial feasibility, iterative testing, and drug safety data, typically in laboratory animals. So now this preclinical development, it is also known as preclinical studies, or it is also known as non-clinical studies. And it is a phase of drug development which starts before the clinical trial process. So as we have already discussed, and the testing is done on animal, okay, and to get more knowledge about the drug safety and its efficacy. Okay. So preclinical studies often involve novel medical devices, prescription medications, and diagnostic, and their primary objective are to establish a beginning safe dose for first in human research and valuable, and evaluate any potential toxicity of the product. Okay, so the preclinical studies, they involve the use of novel medical devices generally. So the preclinical studies not only on drugs, it can be done on medical devices, it can be done on prescription medications, it can be done on diagnostics, with the primary objective that to establish that the drug is, or whatever it is, medical device, it is a drug or it is a diagnostic, it is safe and safe and effective to be used on humans. It does not have any potential toxicity. So companies utilize exaggerated numbers to highlight the dangers of preclinical research, such as the fact that on average only one compound out of 5,000 that enters drug discovery and advances to preclinical development is eventually authorized for use as a medicine. So now there are the companies which generally exaggerate the numbers, and they highlight the numbers that it is very dangerous to conduct a preclinical study. But the fact is that on average, like it is said that from labs and from crores of compounds, there is a one compound, but that is not true. On an average, at least from out of 500 compounds that enters the drug discovery phase, and it advances to preclinical development, which eventually authorized for use as a medicine. Now next is clinical trials. So clinical trials, we all understand that clinical trials is a procedure which is done on humans. So clinical trials are prospective biomedical or behavioral research studies involving human subjects that are intended to provide answers to particular questions about biomedical or behavioral interventions. These include both established and novel treatments, like they can be the novel vaccines, medications, dietary choices, dietary supplements, and medical devices. So clinical studies are not only on drugs, but nowadays clinical studies are also does on the nutraceutical or dietary supplements, and on medical devices, on novel vaccines and medications as well, which are worth further investigation and comparison. Information on doses, safety, and efficacy is produced by clinical trials. They are only carried out following clearance from the ethics committee or health authority in the nation where therapy approval is requested. So now these studies are carried out only when clearance from the ethics committee. So first, like as I told you, that before the preclinical, like after the preclinical studies, the investigational new drug application is filed to the ethics committee, and then after getting approval from the ethics committee, only the clinical trial process can start. These authorities are in charge of assessing the trial's risk-benefit ratio. Their clearance does not imply that the therapy is safe or effective, merely that it may go ahead. So now when the IND is granted and the permission is granted to conduct the clinical trial, so that doesn't mean that the clinical trial, the drug which is used is safe or effective, it means that that approval means that, okay, you can go ahead with clinical studies. Then again, when the clinical studies are completed, again, the drug has to undergo a regulatory process where it is analyzed that or assess the risk benefit ratio, and also like how the drug is progressing, what are the adverse effect and all of the drug, or whether the study is, or whether the drug is fulfilling the objective of the study or not. So that is what the clinical trials are. The next is clinical versus preclinical. So non-clinical evaluation selects a definitive point. So now, unlike clinical evaluation, when there is a definitive point to conduct the study and to start the study, but in non-clinical evaluation, there is no definitive point which is taken into consideration. Toxicity studies is a significant milestone. However, studies are not specifically mandated by the guidelines, but are driven by specific and practical reasoning. So now these toxicity studies, because from the toxicity studies, we'll come to know various toxic effects of the drug, which may damage any organ or which may be life-threatening, or lead to the development of disease which are fatal, such as cancers. It may lead to mutation also, and sometimes they also lead to death of the patient. Okay, so non-clinical studies also contain so these toxicity studies are very significant to be conducted. However, the studies, it is not mandated in any of the guidelines that it is mandatory to conduct the studies, but if you want to conduct these toxicity studies, then they will be driven by some scientific reason or some practical reasoning. The non-clinical studies continue after clinical trials initiation. Some of these include studies that are aimed at supporting late clinical phases, example, the chronic repeated dose toxicity studies, while others are required for marketing approval, example, carcinogenicity studies. So now these non-clinical studies, some non-clinical studies are like chronic repeated toxicity studies and the carcinogenicity studies. So these are the studies which usually requires a lot of time, like from 6 months to 1 year of time, to see the carcinogenic effect or what is the toxicity that develops once the drug is administered continuously for some time, like for a particular period of time, the toxicity can be conducted. So now the non-clinical studies, they continue after the clinical trial initiation, and these studies are aimed at supporting late clinical phases. Example is like chronic. So here the dose of the drug which is used, that is also slightly higher to see the effect of the drug. So now when these carcinogenicity studies and chronic repeated dose toxicity studies are conducted, then simultaneously the clinical trials on phase 1 with very low population can also start. For marketing approval submissions, non-clinical information is traditionally seen as corroborating the clinical evidence. Now, when the sponsor, they submits the CTD dossier and they require that they'll get the permission to market the drug. So for that purpose, they have to submit the non-clinical information along with the clinical evidence. Safety assessment can provide mechanistic explanation of adverse reaction observed in humans, and pharmacokinetic evaluations can inform decision on clinical pharmacology studies like data on carcinogenicity and reproductive toxicity. So now these safety assessments which are done, so they provide you with the explanation of the adverse reactions which are observed in humans. The pharmacokinetic evaluations which are made, which is related to absorption, distribution, metabolism, and excretion, they can inform the decisions or on clinical pharmacology studies like on the carcinogenicity or the data which is available from the carcinogenicity or reproductive toxicity studies. Okay, understood? Is it clear?